I'm going to try and tell you something about how I think fungi colonized hosts and colonized niches more in general. But before I do that, I will tell you briefly where I come from. Not so much academically, but simply the city where I come from, or at least the city where I work. And that city is called Cologne, and it's located in Germany. You can see it here on the complete west side of Cologne, very close to the Dutch border. Actually, I'm born just across the border in the south of the Netherlands, in Maastricht. So very close to the Netherlands and to Belgium. Now, I guess many of you don't know Cologne. Some of you maybe do. The thing that Cologne is very proud of is World Heritage building. Here, the Cathedral of Cologne, the Black Cathedral, very well known. And then Colonians are very proud of something else too, and that's their beer. And their beer they call Kölsch. The rest of Germany is a bit ambiguous. Most Germans are not so fond of Kölsch. On the one hand, because they think the taste is not so special, but actually more important for them, this Kölsch is served, you can't see it really here, but it's served in, for German standards, tiny glasses. So the rest of Germany doesn't like that. Then there is something else that's very well known in Cologne, in Köln, and that Colonians are very proud of. I guess you all know most of the world has 4 seasons in a year. We go from spring to summer to autumn and winter. In Cologne, they have 5 seasons, and the 5th season is the carnival season. And actually, that starts the 11th of November, and it ended last weekend with the big carnival celebrations, as you can see here. And just very quickly, I'm going to flash a photo to show you that I also was present at the carnival this year, 2 days before I entered the plane. But so much about Cologne. Now a little bit about fungi and fungal infections. And I think many of you are familiar with infections and are familiar with host microbe interactions, but I guess also not all of you. So to make sure that we keep that I keep you all on board, I'm going to try and explain you in a view of how I think immune systems and so also plant immune systems work. And so in general, immune systems are systems that can be induced, activated in order to ward off microbial pathogens. And what is critical here is, apart from the fact that it is an inducible system, also the fact that a particular threshold needs to be reached in order to reach a state that we call immunity, which basically means the host is not suffering from the microbial invasion, is not showing disease, if you will. And so all of this starts because plants on the cell surface have receptors, antennae, that detect particular molecules that betray the invasion of microbes. Now, what molecules do plants then typically detect? Well, these are molecules that we here abbreviated MAMs that we call microbe-associated molecular patterns. And as the name then already says, it's a molecular pattern, it's a molecule that is specific to a microbe. It's microbe-associated. And you can also say, basically, this means non-self-recognition. So, for example, fungi have a cell wall composed of chitin. Chitin does not occur in plants, so plants have receptors for chitin. If these receptors detect chitin, immunity is activated, and we get this chitin-triggered immunity. Now, chitin is not the only MAM. Think of bacteria. Bacteria have flagella, composed of building blocks, flagellin proteins. Flagellin doesn't occur in plants. Plants have receptors for flagellin. Same story, we get MAM-triggered immunity. And like this, there is a wealth of microbe-associated molecular patterns. Molecules that occur in microbes do not occur in the host. Now, some people may know this term in a slightly different form, because in the mammalian field, people typically talk about PAMs, pathogen-associated molecular patterns. Some plant pathologists too, but I don't think that's really a proper term, because the molecules are named chitin, flagellin. They're not really pathogen-specific, they're microbe-specific. So I prefer the term MAP. Now, this concept, I show you for plants, but actually for us, it works in the same fashion. Many animals works in the same fashion. So this is believed to be the basis to explain why, despite the fact that there's so many microbes in the environment, plants, when you see them generally, are happy and healthy. However, in some cases, disease does occur, and somehow we need to explain this. And we explain this by the fact that adapted microbes that have evolved to cause disease in some cases secrete molecules that we collectively call effectors. And an important function of these effectors is to suppress this immune response such that we dive below this threshold of immunity and we get to a state that we call effector-triggered susceptibility. And then we get the typical symptoms like here shown on Arabidopsis leaves for Pseudomonas bacteria. We get disease. Now, these effectors is basically a container term of all kinds of molecules that somehow help to reach the state of effector-triggered susceptibility. So what are these effectors? Effectors are molecules secreted by pathogens during attack on their host to modulate the physiology of their hosts and thus enable colonization. And I on purpose say modulate physiology of the host because, yeah, very often we think they suppress immunity. And of course, immunity is part of physiology. But in some cases, we also know effectors do other things. For instance, help to liberate sugars, help to make sure that plants secrete sugars into the environment such that pathogens can take them up. It helps to colonize the host, it modulates physiology, not necessarily immunity. So that's why we define it like this. And I put these pictures here in the bottom to show you, I think, a quite remarkable phenomenon. Namely, these effectors are exploited by all types of pathogens. So by bacteria, shown in the left picture, by fungi and oomycetes. So we already go to 3 different kingdoms, but also animal pathogens, pests of plants make use of effectors. Aphids, through their stylet, don't only suck up phloem sap, but also deliver effectors to suppress immune responses. Nematodes, with their stylet, when they pierce cells in the roots, exactly the same. They use it to suck nutrients from cells, but at the same time also to deliver effectors. So somehow, remarkably, across all these biological kingdoms, these same type of molecules have evolved in order to establish host infections. And this is why we think it's very important to study effectors and then to understand them, because well, we think this tells us something about how microbes, these other pathogens, are able to cause disease on their hosts. And here I show you a picture of a transgenic fungus that has now colonized a plant cell. And it is secreting 2 effectors that are differentially labeled with a different fluorescent label. And so the fungus is Magnaporthi, it's a rice fungus. This is work from the group of Barbara Vaillant. And one effector here stays in the interface between the fungus and the plant host, because now you need to imagine the fungus invaded the plant cell, but it actually doesn't penetrate the plasma membrane. So the plasma membrane of the host is sort of surrounding the fungus. And so that's clearly what we call an apoplastic effector. It stays in the interface between the fungus and the plant. But then the second effector is actually secreted at one focal point from the fungus. So this is still fungus. And then we don't find it anywhere here, but we find it here. And that structure in the cell is the nucleus. So this is an example of an effector that is able not only to enter the cytoplasm of the plant host, but even to find a destination. And in this case, the destination is the nucleus. And so by now we know examples of dozens of effectors and their targets in plant cells. And basically, from all of this collective knowledge, we can say, well, think of any compartment in a plant cell, and there will be particular effectors that target that organelle or that compartment to do something that in the end results in modulation of host physiology, suppression of host immunity, and something that somehow helps the microbe to colonize the host. Now, I will give you one example of a very specific effector activity. And I gave you this example because it will come back a bit later in my talk too, but also because this is an effector that's actually used by many fungi during host colonization. So in my group, we mainly focus on fungal pathogens. And this effector that I'm talking about now is called the lysem effector because it has so-called lysine motifs. We know they bind chitin. And so here you see the interior of, let's say, a tomato leaf, and here you see this fungus growing. This fungus has chitin in the cell wall. I explained to you chitin is a MAMP. I explained to you too that plant cells have receptors for chitin. So once they perceive chitin, they will activate MAMP-triggered immunity. They will activate chitin-triggered immunity. And normally this whole system is supposed to, of course, contain the fungus, to slow it down, to stop it from colonizing. Now, fungi have evolved a trick to deal with this, because what they do, what most fungi do, is they secrete so-called lysem effectors. And these lysem effectors you can see as molecular vacuum cleaners. What they do is they bind and immobilize small chitin fragments, such that these do actually not activate these host immune receptors. And then, of course, the plant or the host doesn't know that these fungal fragments are there, that this fungus is colonizing, and by the time it does realize it's being invaded, it's too late. So I think this is a very, in my eyes, beautiful example of how evolution made that fungal pathogens evolve this mechanism to suppress chitin-triggered immunity in plant hosts. So now I guess you all know a bit what effectors are, what kind of things they can do, where they occur. So this explains disease in particular cases. But then, of course, you also, I guess, all know that this is probably not the end of the story. There are receptors, again, that are able to activate immunity because they recognize effectors for their activities. So we're going to draw another arrow up that leads to so-called effector-triggered immunity. Now, not based on the recognition of MAMPs, but based on the recognition of effectors that evolved to suppress immunity. Now, you can easily imagine, I guess, that these MAMP receptors are widely conserved throughout almost the plant kingdom, whereas these receptors that perceive effectors are much narrower distributed. So they occur in particular lineages of plants. Now, obviously, once this happens, there is again pressure on the microbe, pressure on the pathogen. If the pathogen has the effector, basically it's not able to colonize, it's contained, so that puts pressure on the pathogen to evolve. So pathogens then mutate these effectors that are recognized, lose these effectors that are recognized, or evolve new effectors to again suppress effector-triggered immunity. Examples of all of these are present or can be found in literature. And of course, once that happens, there's pressure on the host again to evolve a new receptor to recognize the variant or to recognize a new effector. And this is an everlasting arms race, typically, between pathogens and their plant hosts. So this is, I think, a bit of context in which a lot of the work that we do also operates. Now, I'm going to talk about a fungal pathogen that we call verticillium. And here you see a video, maybe you recognize these are two seedlings, these are two tomato seedlings. And slowly you will start to see that there is a difference between these two seedlings. One is a control plant, a plant that has not been inoculated with the pathogen. It's the one on the left-hand side. The one on the right-hand side, you can see, starts to look a bit pathetic. So it has been invaded by this verticillium fungus. Verticillium is a soil-borne fungus. It will penetrate roots as soon as it senses root exudates, and then it grows into the xylem vessels. And that's where the fungus then starts to sporulate, and these spores are carried with the sap stream. They get blocked somewhere, they grow a bit, sporulate again, and like that, that whole plant is colonized. And because it colonizes the vasculature, because it colonizes the xylem vessels, it basically blocks water transport. And well, you can see the effect, the development of the plant is severely hampered. Now, this is a very severe case. It's not always that severe, but generally this fungus causes wilt disease. And now this video is obviously about tomato, but verticillium is a pathogen of hundreds of plant hosts. And even individual strains have very wide host ranges. And we think this is therefore also a very interesting pathogen to study. Now, of course, also verticillium obeys to this zigzag scheme that I showed you. And so this story here starts with telling you that there is recognition in some tomato genotypes. These have a resistance gene that's called VE1. And by now, I guess we know that, okay, if there's a resistance gene that activates effector-triggered immunity, there must be an effector that's recognized, and that effector is called AVE1. Now, the effect of this effector is not only to activate effector-triggered immunity, right, to make this arrow go up. From the introduction, I hope you realize that, no, these effectors have a primary function to, first of all, suppress some kind of immune response to help colonization. And this is basically shown in the picture in the bottom because these are tomato plants that do not carry this immune receptor. So they do not do this response and we're still stuck here, you can say. So you see a mock inoculated plant, that one looks pretty happy, healthy. We have the effect of the wild type fungus. And you can clearly see the stunting, maybe not as dramatically as in the video, but clearly that plant is stunted and not as happy and healthy as the mock inoculated. And if we now take this same fungal strain and delete only this effector gene that in other tomato genotypes is recognized by this VE1 immune receptor, we can basically determine what the impact is of that one single effector on the ability to cause disease, basically the impact of this arrow. And I hope you appreciate that that plant is much bigger, much healthier. So this one single effector makes quite a big contribution to disease development. And that's the reason why we were very much interested in finding out what does this effector do? How does it make verticillium so much more aggressive? And that's a story that took us almost 15 years. And the first 10 years of these 15 years, we tried to find all kinds of host targets. We did everything we could. Nothing worked out. Until we realized, well, maybe we have two clues in our hands that we have not fully appreciated, but they tell us something. The one is, based on the scheme that I showed you, I hope you realize that, yeah, it makes sense for a microbe to produce an effector the moment it tries to cause disease on a host, the moment it tries to invade a host. And so what we typically see for effectors is they are strictly implanta-induced, implanta-produced. Now, that's not exactly true for that AV1 effector that we count. Clearly, it's expressed in planta, absolutely. But also in other conditions, we find expression. Now, one condition in the lab is in vitro, but for the biology of verticillium, that's not very interesting, obviously. But another condition that we then started to test is, but what happens if the fungus is not in the plant? That means when the fungus is in the soil. And also then we saw expression of this effector gene. Not immediately expected that the sole or the primary function is to suppress host immunity. The other clue that we did not really appreciate was that we did in these 10 years all kinds of things to try and produce protein in heterologous systems such that we could do biochemical assays. And all these production systems didn't give us any soluble protein. And at one point we started to realize, yeah, well, wait, but maybe these microbes don't want to produce that protein because that protein is harmful. So it's an antimicrobial. And if it's actually an antimicrobial, well, it would make sense why verticillium does also produce it outside the plant host, because maybe even the impact of microbial competition is higher in soil outside the plant than it is inside the plant. So once we put these two clues together, we started testing this hypothesis. And what we did is, so what happens in E. coli if you try to force it to produce antimicrobials, very often either the clone just dies or it will produce it, misfold it, and bring it to the garbage bins of the cell to inclusion bodies. But we can harvest the protein from these inclusion bodies and then in vitro denature it, refold it, and we can actually test on plants that have this immune receptor whether the refolded protein is able to activate immunity, and if so, it's very likely correctly folded. It's what we did. We saw immunity, and now in a nutshell, I'm going to bring you fast forward. So what we then learned is, yes, this effector has antimicrobial activity. It has selective antimicrobial activity. Not all microbes are inhibited, very particular. So here I show you for Bacillus subtilis, a Bacillus bacterium, growth of the bacterium in medium in the red line, and in the presence of this effector gene, a complete flat line, there is no growth at all of this Bacillus bacterium. There is no growth, that makes sense. If we look under the microscope, bacterial cells just completely collapse. It's a cytotoxic protein. Well, this is all in vitro. That doesn't tell us so much. What we then decided to do is microbiota profiling in a plant. And we did this in various plant species, in tomato and in cotton. We inoculated with the wild type fungus and the fungus that lacks only this one effector gene. And what it showed us is that we don't see big changes in the microbiota, but we see some bacteria that disappear as soon as the fungus produces the effector protein. In this case, we saw that sphingommonad bacteria are depleted from the microbiota. And then we started testing what is the role of these sphingommonads, what do they do to verticillium, and basically it showed us two things. It showed us on the one hand that these sphingommonad bacteria are antagonists of verticillium growth. So if you expose verticillium to these sphingommonads, they hamper verticillium growth. And so if we now coat seeds with sphingommonads and then grow tomatoes and then try to infect them with verticillium, on the one hand we see that these sphingommonads protect plants to some degree against verticillium infection. The more sphingommonad bacteria, the lower the infection rate. And on the other hand, we saw that if verticillium now has AV1, this effector, it's able to deplete these sphingommonads, whereas if it doesn't have this effector protein, it's not able to deal with these sphingommonads. So to sort of put that all together in a little bit kind of a simple scheme, basically the bottom line message is we have now here this root system, we have these resting structures of verticillium, they will germinate as soon as they sense root exudates, they will start to penetrate the roots. Obviously that leads to the activation of immune responses. That means activation of typical plant immune responses, but actually we by now also know it also means the recruitment of beneficial bacteria into the microbiota. All of this meant to slow down or to stop this microbial invasion. And so these microbiota, the beneficial characteristics of the microbiota, it's aimed at trying to suppress these invading pathogens. But then verticillium, at least when it comes to this one effector that I described, produces an effector that's able to target specific microbiota members, namely those that act as antagonists, sphingommonad bacteria. And as a consequence of this, it of course has better abilities to colonize this plant host. So when we found this, we were quite surprised and we got the question frequently, okay, but is this a general thing or is this just a weird effector that happens to have this activity, but actually this is something you rarely find? And we are now convinced, no, this is not something rare. This is something extremely common because actually we have now characterized more than a handful of effectors that display such types of activities in verticillium. And the bottom line message of this is we have all kinds of different effectors. They're expressed in life stage dependent manners, some in the soil, some strictly in the plant, some in the soil and the plant, some only in early stages, some only in late stages. They all have selective antimicrobial activity. Some are antifungal, others are antibacterial. And so basically, I think that adds to it a sort of generally emerging picture where we realize that, of course, plant immune systems are there to restrain pathogen colonization. But at the same time, plants are able to shape and to sculpt a microbiota. And that microbiota also has a function to try and minimize the impact of detrimental microbes. And as such, you can see this as a second layer of the immune system or an additional layer of the immune system. Now, if you appreciate that during evolution, pathogens have evolved to target critical nodes of host immune systems, yeah, then it's not so surprising that if they do this with effectors, that effectors do not only target the host immune system itself, but also target the host-associated microbiota in order to establish host colonization. Now, for verticillium, I think we have pretty well established this now, and actually I'm very happy to see that in more and more cases for different pathogens from different hosts, effectors are now found and reported to be able to manipulate host microbiota. And so basically with that, I think it's pretty obvious that this phenomenon that we found for verticillium is not something verticillium specific, but is something that is widely spread among plant pathogens. And this basically, I think, needs to bring us back to this definition of effectors too, because by now, I think this phrase that I had in this definition saying that effectors target host physiology or host immunity, well, I don't think we should really say this anymore. I think effectors are molecules secreted by pathogens that basically do anything as long as it helps host colonization. So now this phenomenon is widespread among plant pathogens. Actually, I think even more widespread. And for us, that triggered the question, where do these effectors actually come from? Especially because we started to see that some of these effectors seem to be ancient molecules, very widespread. And so we have started to work on the hypothesis that perhaps this is a trait, the use of effectors to manipulate the microbiota, that evolved in ancestor fungi far before they were plants of, they were pathogens of plants or pathogens of other organisms. And so we have proposed in a review a couple of years ago that in these ancestral environments, aquatic environments, terrestrial environments, ancestor fungi evolved in fierce competition with other microbes for very scarce carbon sources. That's where they evolved a lot of these tools to combat each other. And later during evolution, when fungi evolved to become associated with plants, either as pathogens or as other types of symbionts, or with animals, mammals, you name it, or even in other types of associations, such as lichens, that during this co-evolution, these organisms co-opted these molecules to serve functions during the colonization of these different niches. And that's an hypothesis we now really try to work on and try to demonstrate. So the theory now also is that many of the factors, as we know them, actually evolved from these ancient antimicrobials. Now, in order to further develop this hypothesis, it is crucial that we're able to recognize antimicrobial effector proteins. And until now, this is a bit of a challenge, because how do you recognize them? Well, one way is, of course, to produce effectors one by one and test them, but that is very laborious. So we decided to try and set up a machine learning tool that would be able to recognize effectors that display antimicrobial activities. And so to do that, we composed a training set. We went in literature and we took any protein in the size range of an effector, let's say up to maximum 1,000 amino acids, that was characterized to be an antimicrobial, to have bona fide antimicrobial activity. And that results currently in a set of about 150 proteins. And in the bottom, you can see that some of them are derived from animals, others are derived from plants, some from fungi, a diversity of organisms. For us, it's not so important because the main trait that we're interested in is not where it comes from, but what is the activity it has. Is it an antimicrobial? And then we try to extract all kinds of features from these proteins. So you can see here, I mean, it's very busy, but the take-home messages, we try to extract all kinds of things from the primary sequence, but then we also predict structures for all of them, try to predict, to take out anything that we could from all these structures and feed it into the machine learning tool. And in the end resulted in a classifier that on the one hand, for a given structure, tells us, is this an antimicrobial, yes or no? And if it is, what is the probability that it is an antimicrobial? And so we have done tests. I'm not going to show you. I had a QR code there. If you're interested, there's a bioarchive. There it's all reported. Our conclusion is it's a very reliable predictor. And now we use this predictor and we ask the question, what is the occurrence and conservation of antimicrobial effectors in the fungal kingdom? And step one was, okay, let's go to our favorite fungus. Our favorite fungus is Verticillium, has been introduced as a plant pathogen, and it's an ascomycete fungus. But then we added two other fungi too. We added Coprinopsis, a saprotroph, different lifestyle, a basidiomycete, different branch of the fungal kingdom. And we added Rhizophagus, a mycorrhizal fungus, yet another lifestyle, plant associated but mutualist from the Glomerulomycota. And now we focus on all proteins that are secreted. And in this secretome, we know there are carbohydrate active enzymes, K-zymes, but we put them aside. We don't think they are immediately the most interesting, and also it's very difficult for a single one to say whether it's truly an antimicrobial, yes. So we focus on all the rest, because there we think we have more difficulties to determine whether it's a probable antimicrobial activity. And all this rest, we then ran through our predictor, and actually, irrespective of the species, irrespective of which part of the fungal phylogeny, and irrespective of the lifestyle, we find that the majority of this secretome, the majority of what is not a carbohydrate active enzyme, is predicted to be an antimicrobial. And actually, with the majority, the far majority of that again, with a very high probability. And to us, that was an enormous surprise. So that basically means a single fungus produces hundreds of these antimicrobials. So then we decided to take this a step further. So now we go to 150 different fungi, indicated here in the left, spread across fungal phylogeny, spread across fungal lifestyles. And for these 150 fungi, we determined ortho groups, so conserved, secreted effectors, you can say, or secreted proteins that are conserved among these 150 fungi. The most conserved on the left, and the further we go to the right, we see a drop in this conservation. But even for the last one, I think it's pretty clear, still pretty conserved throughout these 150 fungi. And then for each of these ortho groups, we predicted, are these now antimicrobials, yes or no? And that's displayed in the bottom. When it's green, it's a carbohydrate active enzyme. When it's white, it's not an antimicrobial. But when it's red, it is an antimicrobial. And I think also here it shows that the majority of these are actually antimicrobials. Again, quite surprising. So if that is really true, then that means that many of these effectors are antimicrobials. And if that is true, then we slowly need to think about the fact that maybe the typical effectors as we knew them that suppress host immunity, actually, like I proposed, derive from antimicrobials. And now I go back for a minute to these lysine effectors, and I told you what they do. They very clearly suppress chitin-triggered immunity in plant hosts. Very clearly interfere in host immune responses. Now, these lysine effectors are also very widely conserved among fungi. So that was for us an obvious one to start testing, because we would never have anticipated that these are antimicrobials. And here I show you for 3 different lysine effectors tested on a panel of bacteria, and that indeed, they seem to display antimicrobial activity, here shown in red. Not all bacteria, again, selective activity. mostly gram-positives in this case. I forgot to say, so here are these 150 fungi where we display the phylogeny also of the lysozyme factors that we find, and on the outer ring in red indicate for each and every individual lysozyme factor whether it's predicted to be an antimicrobial, yes or no. So for me, at least, the take-home message is, yeah, well, most of these indeed are antimicrobials. So that's just one example, but we would like to have stronger evidence for this, more evidence for this. Now, what we, of course, can do is try to find new effectors, and on the one hand, determine whether it's an antimicrobial, and on the other hand, try to find the host target of such an effector, but that is quite complicated. So we thought, we can maybe do a smarter strategy, and the smarter strategy is make use of the FABI, the pathogen-host interaction database, where a lot of information is assembled on molecules, genes that are crucial for host microbe interactions. It also includes effectors. So in this database, we find 130 fungal effectors, and we decided to make use of this by saying, okay, we're going to look at those that, of course, are fungal effectors, but we want them to be widely conserved because that means their origin is ancient. So we looked at homologs, widely conserved among these 150. We said present in minimum 100 of these. We looked at those for which we can predict the structure very well, and then we predicted antimicrobial activity. And here you now can see a set of effectors that we identified like this, that are very widespread, that are predicted to be antimicrobial with a very high probability. And those we decided to target and to produce and to determine their effects. Now I'm going to give you one example, but we have now similar examples for several of them. So the one example is actually this one here in the bottom. 424Y is the name. It has been studied by different groups, and all of them show that this effector suppresses host immunity because it goes into host cells and it manipulates salicylic acid metabolism. So we're pretty convinced if multiple groups report this, then it must probably be robust. It's a glycoside hydrolase. It has a homology to glycoside hydrolases, but it localizes actually in host nuclei and activates the immune signal. Homologs in many of the 150 fungi, actually the far majority is predicted to be antimicrobial. And when we test this, we indeed see this. So activity on bacteria, selective activity, the bottom bacterium is not affected, but the top bacterium is clearly affected because the green line is now growth of the bacterium in presence of the effector protein. But now this one is not only antibacterial, it's also antifungal. Here, two fungi, a yeast and a filamentous fungus that is clearly inhibited by this effector protein. And now, all of this in vitro, somewhat artificial. We also try to show these effects now in planta. And here we make use of a gnotobiotic system where we'll also be able to test plants in absence of a microbiota. And interestingly, now look at these mock-inoculated plants and the occurrence of, in this case, verticillium. We look at the verticillium homolog. We see an impact of the verticillium infection with the wild type, and we see a same effect when we inoculate the plants, not with the wild type fungus, but the fungus that has deleted for only this effector gene, this 424 effector gene. In sterile conditions, we don't see any difference in colonization. But when we then go to conditions where plants have a microbiota, again, the mock-inoculated plant is healthy, actually a bit bigger than the sterile plant, we know. The impact of verticillium infection also very clear. But if we now delete this gene from verticillium and do the inoculations, the disease development is clearly reduced, is less. And so that tells us that indeed, this one effector that has been shown to be crucial for suppression or modulation of host immunity actually has a function also to manipulate the host microbiota. And so with this, I've come to the end, and I hope I convinced you of some of the take-home messages that I would like you to appreciate. First of all, I think microbiota manipulation is incredibly important for host colonization. I showed you that effectors with antimicrobial activity are widely found across the fungal kingdom. We, of course, study them in the context of plant pathogenicity, but other fungi, non-pathogens, have them too. And there they perform similar roles in manipulation of microbiota, in that case, not host microbiota, obviously, but microbiota in their environment. We think that many of these antimicrobial effectors actually have a very ancient origin. They're highly conserved. And we get more and more evidence that some of these effectors evolved completely new functions, often even while retaining the original antimicrobial activity. But they obtain new functions, for instance, to suppress host immune responses. And for us now, this also starts to trigger a completely new thinking. I started explaining you this scheme with these arrows going up and down, explaining the arms race between pathogens and hosts. And now I think there is an arms race, a very similar arms race that takes place that we have actually completely ignored. Because while focusing on the interaction between the pathogen and the host, actually this pathogen is also busy fighting a battle with these host-associated microbial communities. And I'm very much convinced that also there, these arms race type interactions of developing tools, making escape routes, and finding molecules to suppress things and so on, similarly takes place. So, with that, I'm going to show you the final slide, which is perhaps the most important, the acknowledgements. A picture of my group in the greenhouse at the University of Cologne. All the names of the group members are there because obviously they are the people that did the very hard work. And with this, I'm going to stop and thank you very much for your attention.
Thank you for indeed very exciting and thought-provoking, as Roselia was saying at the beginning. Really challenging, I think, how we're thinking about plant pathology in a general way. So really appreciate that. Let me see if there are questions from the floor. And Monet, if you could just help me check if there are questions online.
Very nice. Thank you very much. I enjoyed it a lot.
Thank you.
I have got two questions that are kind of linked. So when a plant gets infected by a pathogen, it has its own immune response. And this then basically has also a general antimicrobial activity towards all the endophytes or whatever you want to call these organisms that sit inside the plant these days. So what do you think is more important in suppressing them? So we often see lower levels of microbial diversity in plants that have actually been infected by a pathogen. Is the host response or is the pathogen effectors, which one would be the most important one in this relationship?
That is an extremely difficult question to answer. But also one of the questions that intrigues us a lot. My feeling is it's very difficult to answer this in general. However, a plant immune response, I think, evolved such that it targets a diversity of microbes because the potential pathogens, is a wealth of microbes. What we tend to see, which initially surprised me, but the more and more I think about it, the less I think it's surprising. Many of the effectors that we characterize as antimicrobials have very specific activities. And so until now, when we find in the microbiota profiling that a particular effector suppresses a particular microbe, and we start to test the impact of that microbe on the pathogen, we always find that it's an antagonist. And so my interpretation here is that many of these effectors are quite sophisticated molecules that do not target a broad spectrum of microbes, but target specifically those in the host-associated microbiota, from which the pathogen suffers, the antagonists. And this is a product of a very long coevolution. Now, for example, verticillium, I imagine that the number of true antagonists is, I would almost say somewhat limited. And so in a host microbiota, it will pick out these very specific members and target them. Whereas I guess the effect of the plant immune system may be a bit more brute force, because this is a survival strategy, right? The moment that this infection is happening, the host will need to clear it somehow. Otherwise, it may not survive. So my prediction would be the impact of the host antimicrobials is on the total microbiota is bigger.
Okay, in this case, you actually did, in your second part, answer the question I wanted to know, how broad spectrum these things are, which you answered.
Thank you very much for an intriguing presentation. So mine is a very simple question. So in the arms race between the pathogen, the fungal pathogen, and the microbiota, so who's winning the race?
That's extremely difficult to say. I don't think at the end of the day, someone wins. It's the same as the arms race between the plant and the microbe. I mean, I don't think we will reach a state where one wins. This is just an everlasting race that goes on and on and on. So far, well, I mean, verticillium is still around, so it's not losing at least, but it's difficult to say who wins. I think it also depends very much, and here we don't have knowledge. In a given environment, in a given context, maybe the microbiota wins and we don't see verticillium there anymore. But of course, that requires knowledge on knowing that it has been there before. We don't have that.
Right, I see questions here in the room, but I also see Londiwe has a question online. Londiwe, go ahead, please. Londiwe, floor is yours.
He's still muted.
You're still muted, Londiwe.
To allow the unmute function. Nice.
Okay, can you hear us now?
Yes, we can hear you.
It's Volmery, actually, not Londiwe.
Just a second, we're not hearing you very clearly.
We are from Stark East. It's Volmery, but not.
Go ahead, Volmery.
Can you hear me?
Yes, a little bit in the distance, but we can make it out, yes.
Sorry, we've got a thunderstorm here in Kempton Park.
That's much better.
Yes, but I've got two questions. The one is, does that mean, or does that relate to induced resistance that you get when one pathogen infects a plant, that it can then induce resistance against other pathogens? Is this linked to your effector response?
So, well, the short answer is we don't know. We have not looked at this. I mean, the whole idea of induced resistance obviously is that there is a trigger that then, I mean, the induced resistance phenomenon clearly is a plant phenomenon, right? Yeah, I don't know very well. Well, let's put it like this. Again, the short answer is we don't really have evidence for this, but we do know, for instance, that there are effectors produced by verticillium 2 that specifically target the recruitment of Pseudomonas bacteria. And these Pseudomonas bacteria, we know, play an important role in these induced resistance phenomena. So I would not be completely surprised if, yes, they have an impact, but we would have to specifically look at that to see whether this is absolutely true, whether we can solidify this. We have not done this. Nice suggestion.
Does that mean
you have another question?
Yes, to follow up on that, does that mean if you can, that the antimicrobials produced by verticillium could actually protect the plant against other pathogens?
Yeah, that's,
Is actually what I'm thinking.
um, hmm. Theoretically, yes, but it will be a very delicate thing because we now start to realize that all these different antimicrobials, they all have their activity spectra, and they all do something in this plant microbiota. So yes, of course, definitely you will have antimicrobial effectors that in principle would be able to suppress other pathogens. But the question, of course, is, but what does it do to the host microbiota, and to what extent does it actually interfere with the host microbiota so that you get other problems, so that you open the door again for other types of pathogens? So theoretically, yes, but it may be a tricky business.
Thank you very much,
Okay.
Lucy.
I will email you some other questions. Thank you, Bart.
Thank you.
Thank you. That's a really nice seminar, really interesting topic. So as we were talking, I was thinking about phytopathogenic bacteria or bacteria in general. Historically, the type 3 typically had that same effect where we thought of effectors targeting host immunity, full stop.
Yeah.
And then came along the type 6,
Six.
which particularly in phytopathogenic bacteria had a lot of antibacterial or anti-other microbes.
Yeah.
My question is, you are seeing some of the effectors of the fungal pathogen that have dual role.
Yeah.
Suppressing immunity and sometimes having antimicrobial activity. Do you see a kind of motif that would indicate which way this effectors are going to go?
Yeah.
Do you think if we go back to some of the type 3 secreted effectors, we're going to see a lot, I mean, depending on how we design experiments, we're going to see maybe similar to what you're seeing with these fungal effectors. And final question, nematodes, I didn't see that on your list of predicted antimicrobials. So those 3 questions. I hope you
To go with the last one, we haven't looked at nematodes. I mean, we realize now we look at the effect of effectors on bacteria, on fungi, but we should go broader. We should also go to protists, to amoeba, to nematodes, other types of organisms we have not tested. And in the same fashion, I think, I would not be surprised if nematodes also make use of these antimicrobials. I mean, some of them we know, because, for instance, defensin molecules, widely spread, I think, also occur in nematodes as antimicrobials. So will also be exploited. The type 3, type 6 is extremely interesting. I mean, I lack a bit of background, and so I'm not exactly sure how and when these systems evolved. But intuitively, I first of all would think about type 2 in bacteria. Now, for the fungi, for the story I told you about, it's very important to realize that I talk about those effectors that are widely conserved, right? Because it's clear all organisms, and here I think perhaps type 3 in bacteria, all these fungi also acquired lineage-specific effectors. Now, these evolved much later, and to some extent, they may have evolved from the same pool of effectors that I talk about, but to some extent, they will also be new inventions. So for these new inventions, of course, it doesn't have to start with an antimicrobial, because we think this origin of these antimicrobial effectors is so long ago. I think intuitively, for the bacteria, these effectors that are secreted in the environment and that at the same time play a role in the modulation of host colonization, I mean, these have the highest potential of displaying these 2 functions. I mean, the type 3 secretion directs this effector directly in the cytoplasm. I mean, there an antimicrobial does not have much of a function. I mean, there's very few microbes that actually occur there. But then we need to think about phytoplasmas, for instance, or viruses. But I think that's a bit less likely that the microbe has to manipulate these in order to establish a niche, because there is already this physical separation. So I believe the type 2 effectors of bacteria would be the effectors where I would be intrigued to see, do you find effectors with dual roles? Especially also because I think perhaps the group that is the most likely to, let's say, the earliest adopt this double function is the group of effectors that we immediately put aside, namely the carbohydrate active enzymes, the cell wall degrading enzymes. Because there, I think, perhaps it doesn't take so much to make that the activity spectrum of a carbohydrate active enzyme slightly changes, and now all of a sudden also attacks a microbial cell wall in addition to a plant cell wall, or the other way around. So that's where I would expect to find these functions, perhaps too.
So, thanks. So it's fascinating. So I've got a question about specificity and the broad spectrum, because I often think of defensins which are very, very broad spectrum. So in these conserved effectors, do you get any which are broad spectrum, or they're all very specific? And the ones which are specific for a particular bacteria, what is the mechanism? Is it a protein-protein interaction which is making the specificity? Because you could just punch a hole in the membrane, and any membrane may be punched.
So first of all, with respect to the activity spectrum, we don't really have yet a good answer why. But what we tend to see is that the effect on bacteria is really narrow spectrum for all the effectors we characterize so far, the ones that have antibacterial activity. When they have antifungal activity, the activity spectrum tends to be much broader. Now, there is some literature that suggests that the fungal microbiota of a given plant is more variable and anyway is less extensive than the bacterial microbiota. So whether that has something to do with it, I don't know. I think it's too early. We need to start looking into this, but at least it's an intriguing phenomenon, I think. Whenever it's antibacterial, the activity spectrum is very low. And my explanation for this is, so first of all, when I think about fungi and antimicrobials, I think about antibiotics. I think about secondary metabolites. And I think these very often have broad spectrum activities. But then I think a fungal symbiont of a plant, whether that's a mutualist or a pathogen or an endophile, I don't think it's in their interest to turn a complete microbiota upside down. I think that may lead to a stressed host and maybe actually to the activation of immune responses and all of this. So this is my hypothesis why I believe that many of these antimicrobial effector proteins target the bacterial component of the microbiota in a subtle manner with very narrow activity spectra. I think I covered 2 questions, but there was the third.
Because we can speak about this later, because it also worries me that, if you think this is very ancient evolution, you'd think that in the very ancient times it would be in broad spectrum which would evolve first, and then it must have got very, very specific.
Absolutely.
later.
I think the narrower activity spectrum evolved later and is the product of this very long coevolution, because otherwise I also cannot explain why you now need dozens, hundreds of these. I mean, if they all would be broad spectrum, you have a lot of redundancy. But the mechanism, that was the third one I remember. So we're looking into this. And I think the short answer here is, well, we see typical mechanisms that are known for many antimicrobial proteins. So yes, they make holes or pores, they bind cell wall components, all of the typical mechanisms. But of course, this is a slow process, right? I mean, we go effector by effector and we don't have many mechanisms yet. But what we tend to see is, despite the fact that the mechanism is so general, For instance, AV1, I mean, we have now good evidence that it binds LTA, lipoteichoic acids. It has charge patches that then come close to the plasma membrane, and it just causes membrane leakage. I mean, based on that sum up, the activity spectrum should be high, but it's not. And I think that illustrates that, again, many microbes have evolved such that they have modifications on their LTA, or that they have other ways to make that, you know, most of these antimicrobials do not immediately have an effect. And I think this is exactly this co-evolution thing where I believe that despite these very general mechanisms, yeah, the pathogens have to produce, or fungi now have to produce a whole battery of them because, yeah, in this very long arms race, of course, a lot of counter defense and of course a lot of self-defense has evolved. So this is why I think in the end we end up with these very, very narrow activity spectra. Yes, very likely from originally being very broad spectrum antimicrobials. I agree very much with you. Yeah. Thanks.
Last question.
Other questions?
We might just stay here a long time. But that's an excellent talk. Thank you very much. It was so stimulating. Regarding the specificity toward antimicrobial that we propose to be evolved after. And in one example, you show that I forgot the bacteria name, that it actually promotes the growth of the plant when we do the sterilizing. So does it suggest that this fungus specifically targets those with a good relationship with plant or not? And if so, what in for the plant in terms of evolution to keep those in relationship with the beneficial microbe in check? So do we know what the exchange is? And then I might have another follow-up question.
So first of all,
oh no.
First of all, the effect that I briefly pointed out but didn't explain properly is the effect that if you grow plants under sterile conditions in the absence of a microbiota, they will actually develop less than when you grow them in the presence of a microbiota. And that simply tells us plants also need a microbiota in order to fully develop, to fully mature an immune system, but also to grow better. So yes, it is very clear, plants depend on a microbiota, and within that microbiota, also on typical microbes for these beneficial functions. I mean, I guess we slowly start to learn more and more about this, but I think reality is for many of the microbes, we don't even know what their contribution is and how exactly they help plants. But that they do, I think is very evident. So yeah, now you get this sort of co-evolutionary trajectories, right? I mean, yes, plants evolve to sculpt these microbiota, they evolve to particularly make sure they cooperate with the helpers, whereas, of course, pathogens try to evolve such that they interfere with this, that they interfere with the microbes from which they suffer the most. And I mean, yeah, I think sorry for the stuttering. I'm a Humboldt professor now in Cologne, and I don't know how many of you know who Humboldt was. But Humboldt is actually, we have to place him before Darwin, and there is a beautiful biography. I typically don't enjoy reading biographies, but this one I devour, by Andrea Wolff, where she describes the life of Humboldt and the impact of Humboldt. By now I'm convinced Darwin was inspired by Humboldt, but it's actually Humboldt who was the bigger scientist. And he is the father of ecology. And he basically said, and I think that's the bottom line sentence I want to say to your question. He basically came with this proposition, everything is connected. That's, of course, the basis of ecology, but also here, I think very much, this whole idea of plant pathogen interactions is a plant and a pathogen, and now we can explain everything if we study this and if we unravel the molecular details. No, that's nonsense. This microbiota is part, well, actually the pathogen is part of the microbiota, but the microbiota is an important component in this whole system too. And without understanding this, we don't understand plant-microbe interactions. And so, yes, there's all kinds of interactions in this very complex, together the plant, the richness of the microbiota with all its interactions again, because also there you have competition, cooperation, antagonism, you name it. And then there is the pathogen that tries to invade this and again starts to interact with all of that. So I think it's incredibly complex.
Right, we'll keep going for a little while. There's a question. Alan, is that the one? How can this information be used for creating durable
resistance? Yeah, that's of course also something we try to think about a lot. And now what? I mean, I'm quite convinced that we make things more complex because clearly there's thoughts about how we can perhaps give plants a healthy microbiota or work with biocontrol. But how is this going to work if now we actually realize that pathogens evolve tools to disarm beneficial microbes? So one of the things we're thinking about is if we now know crucial tools, for instance, of verticillium, we know that AV1 is very important. We know that it uses AV1 to suppress sphingomonads. We know that sphingomonads, in principle, are beneficial bacteria, can suppress infection and help plants very well. Well, now we can think about maybe using AV1 to train sphingomonads to basically select sphingomonad strains that are insensitive to AV1. So if you know crucial effectors and crucial activities, perhaps you can then use that to select for bacterial strains that are insensitive. And maybe those can be used for biocontrol purposes. But this, until now, is all theory. We have not tested whether this can be done.
Fascinating. All right, I think I saw 3 hands, and I'm only going to allow those 3. Let's see how we go. If we can stick to 1 question each, and I'll start at the back and then move forward.
Hello.
Hello.
I was wondering about the dual role. So if the antimicrobial function was the ancestral one, so would that mean we'd see similar versions of these antimicrobial effectors in the non-pathogenic relatives?
That's it. Perfect. Exactly. Yes, that's exactly the thought. So we believe, and you're fully right, I didn't point that out. So yes, we believe the antimicrobial function is the ancestral function. We believe, indeed, that is probably the function for most of these 150 fungi. But then for some fungi, for instance, verticillium as a pathogen, it used this antimicrobial that it had to manipulate things in the environment to then slowly evolve an additional function. And I think this is likely for several reasons, but one is an important one too that I did not specifically point out later on. The fact that all these also pathogens, of course, I mean, they're not in the plant forever. I mean, there's some exceptions, but the majority not. The majority is, at one point going back into the environment again, and very often for plant pathogens, for instance, that is back in the soil again. And so, yes, these ancestral functions are likely to function there. For lysama factors, we're now looking into this. So we're now looking into non-pathogens because non-pathogens have them too. They cannot have them to suppress chitin-triggered immunity. So we think they have them to manipulate soil microbiota. So absolutely right.
That was a great talk, Prof. Thomma. So I wanted to ask, in the aspect of pathogenic fungi, within your study, has there ever been a focus area in terms of the secondary metabolites that this pathogenic fungi could produce that will aid them in terms of their infection process or infecting their host better?
Yeah, that's a great thought. We have not looked at this. I have explained a bit about this. We have tried to think very much about why do these fungi evolve these antimicrobial proteins, knowing that many fungi have these secondary metabolite type antimicrobials. We have not looked at this because for us, the main pathogen that we are interested in is verticillium, and verticillium does not make many secondary metabolites. It has a couple of clusters, some of course we also know are active, the melanin cluster, for instance. It has some others, but actually we don't have any evidence for implanta expression of these classes. So we think for verticillium, these secondary metabolites do not play a role. But of course, I know in other fungi, yes, they actually play a role. Now I'm not so much aware of studies that really have looked at, microbiota manipulation and the potential impact of microbiota manipulation in the plant host and the contribution of these secondary metabolites to it. But now I go a little bit back to what I was saying earlier. We believe that these proteins have evolved to more specifically manipulate the microbiota. As far as my knowledge on secondary metabolite type molecules goes, I think quite some of them are actually produced by fungi that, for instance, also have cytotoxic effects, cause necrosis and so on. And the moment that is going to happen, it's very difficult to disentangle what is now a direct effect of such a secondary metabolite on microbiota manipulation versus, well, but now we have dying cells and so on, and that also has an effect on microbiota manipulation. So I think this is a very important question, but it's not easy to tackle in a plant host in an infection. So I think we need to think about other ways to address that question specifically.
Thank you, Bart. You showed that Penicillium also has some anti-fungal effect.
Yeah.
So how does it protect itself, and how does it the same process that the surviving fungi has the same process that Penicillium has?
Yeah. This is also one scratch our head. So I can give you one example, and that is clear-cut. One of these antimicrobials actually is a defensin. It's produced, it has very high antifungal activity, but it's produced the moment the fungus, at the end of the life cycle of the plant, starts to produce microsclerotia. It is produced by those cells that develop into microsclerotia. And what we see is microsclerotia are insensitive, and here I'm going to say melanin and all of that. And the rest of hyphae and spores and so on are very sensitive. But that's not a problem. The fungus doesn't need to protect itself because actually the hyphae are going to die, spores are going to die, the microsclerotia are the only structures that survive. So there it's pretty clear, I would say. I have a good round story. But then for several other ones, we don't know. It's clear they have antifungal effects. It's clear that verticillium is sensitive. But somehow, it works. And I think there's a couple of things we can speculate about. One is, of course, we have very little knowledge on where exactly are these proteins produced. And so my hypothesis would be there's very specific sites where they are produced. We base expression profiles on RNA-seq, where you just grind up plant tissue and then you look at expression, but it doesn't mean that all hyphae in that infection site along the whole hyphae produce that antimicrobial. Now, verticillium is a silent pathogen, so here I imagine that perhaps it's secreted at the tips of the fungus. With the sap stream, it's immediately going to be diluted and going to be carried away from the fungus, and like that, it doesn't need to protect us. But we don't know. At the same time, it's perhaps also not unlikely that there are self-defense mechanisms, but perhaps, of course, the sensitivity we test in vitro. When we grow the fungus, we add the protein. Maybe these self-defense mechanisms are only expressed when the fungus naturally expresses the protein while it colonizes the plant host and at the same time secretes a self-defense molecule or something like this. But this is all speculation. We don't have evidence for this.
Yeah. One more online, right? I think this is the last one. Miriam, thank you for the nice presentation. Do you think this mechanism is also used by fungal endophytes? You also think this effectors can influence antimicrobial activity against fungal endophytes or only bacteria?
Well, the short answer is yes. So the bottom line conclusion that we have drawn, and now I'm going to talk about the majority of fungi, maybe, the truly obligate biotrophs, maybe there's a little bit of an exception. But the all other fungi, I would now say, all these 100 fungi that we looked at, we see the same patterns. So the far majority of the secreted effectors display antimicrobial activity, irrespective of lifestyle. So mutualists do this, endophytes do this, pathogens of animals do this, plant pathogens do this. I mean, of course, that doesn't say that that activity is relevant in each and every condition, but I do believe that the potential is there in all these fungi. Absolutely. Actually, I'm wondering, I'm thinking that perhaps we even underestimate. And the reason for this is twofold. One is we started off with our machine learning predictor. And machine learning predictor is based on only 125 characterized antimicrobials that were, published in literature. I mean, if you look at this list of 125, I mean, that's a bit of a biased list. For instance, many defensins are there, and we try to sieve a bit to not over-represent. But of course, I mean, many of these antimicrobials have been identified based on homology. And so I'm very much convinced that what went into the machine learning predictor is a sort of biased pool of antimicrobials. We're now identifying new ones, and it's a bit too early, but once we have a decent set, we're going back and we're going to optimize this machine learning predictor. And I wouldn't be surprised if it tells us that many more of these effectors are antimicrobial. The other thing is, if you go in animal and mammalian antimicrobials, then actually there's a lot of literature on antimicrobial, not proteins, but peptides, small ones. Now I talk about, up to 50 amino acids. These proteins in fungal genome annotation, we basically toss them out because we say these are small genes, these are probably not genes we don't even consider. I don't have any reason to believe that fungi do not make antimicrobial peptides because, bacteria make them, plants make them, animals make them, humans make them, fungi are going to make them too. That's the whole pool of antimicrobials we didn't even think about, we didn't even address. We don't, here when I talk about these effectors, they're not even comprising these antimicrobial peptides. So I'm very much convinced that the pool of these molecules that have antimicrobial activities is even far more than we appreciate.
Bart, I'm going to end it there.